Thank you very much for the organizing experience. It's really nice to be here. It's my first time in the aging meeting, and it's just so nice to be exposed to many different topics. So, as you know, the field of, let me just see if it's working. The field of stem cells has been an integral part of talking about aging and immortality, and this is in this kind of comic immortality cover of the New York Times that, you know, the aging population wants to have better quality of life and live longer, not only when we are sick. Finding organs for replacements and cells are an integral part of the aging research and field and improving quality of life. And that's where, of course, early embryology and developmental potency and the fact that we can derive embryonic stem cells from embryo became a very promising cell type and avenue in this research. And of course, iPS cells and the discovery of Yamanaka made things more exciting because we can take a skin cell or blood cell from anyone and any age and make iPS cells and use really, this is what we showed 15 years ago. For example, in these mice with the mutation sickle cell anemia, we can take a skin cell, make an iPS cell, correct the mutation, and of course we have to differentiate something like a bone marrow, and I'll talk about this later. And we showed at the time we can get 80% recovery in mice. But of course, since then, I want to say, I think it's been quite disappointing because I think as a field, we haven't made enough progress to translating this in humans. And we've been able to solve going backwards and reprogramming, but differentiation with programming has proven a very difficult task. And we're trying to mimic embryogenesis to devise differentiation protocols that are very good. But the problem is that we don't understand embryogenesis, particularly in humans, sufficiently. And I'll talk about that soon. But first, if we want to understand what are pluripotent cells, the type of the conceptual level, we must understand that pluripotent cells and pluripotency is a transient state in development. It's unlike adult stem cells in the blood or the skin where we have them throughout our life. This is a synthesis that exists for 1 and 2 days in development, but we can grow them in the dish by giving them the right cytokines. But this is an artifact. It's a very useful artifact that we would like to use for replacement. But as a field, we understand that pluripotency is more complex and it comes in different flavors. And we have earlier stages of pluripotency, which are kind of the pre-implantation, more potent. We call them naive stem cells. A lot of characteristics I'll not get into. And then afterwards, after implantations, we get a configuration which is called primed pluripotency, which are more advanced. They're not differentiated yet. They're about to differentiate, but they have very different characteristics. And this is very important. And for years, this has been also not known whether this is only a mouse phenomenon or is this existing only also in humans. And do these early multipotent, naive pluripotent cells exist in humans? I'll just briefly show you that years of research from our groups and others, we realized that a major divider between these type of stem cells is that they respond very differently, for example, to blocking FGF-ERK signaling. Naive pluripotent cells prosper and are stable when we block this signaling, and that's a major divider. And therefore, over the years, us and other groups have now devised conditions to grow alternative human naive pluripotent cells, which are more developmentally earlier in times. And of course, conflicts of interest to declare here. Some of these medias are commercialized, but also feel free to try them and support my mortgage. But after all of this, you would ask, who cares? Both are stem cells, both do it the same. And I want to show you an example that it really matters. If you look at the work from Saito in Japan, who did this in mice, and us with Azim Sourani, who did this in humans, scientists have been trying to make germ cells, which are the progenitors of sperm and egg from stem cells, and they haven't been successful. But the key to success eventually by both teams was that they had to start with these naive stem cells, first of all, and then mimic development, go through primed stem cell for 2 days, and then give the cytokine BMP4, and if you do the sequence of it again, you get authentic PGCs that can make sperm and egg if you put them inside the ovary or the testis. So what this experiment shows really that the starting material matters critically whether it will succeed or fail. But we still don't understand exactly why do we have to start with naive cells? Why is priming needed, and why does it exist in multiple species as we now know? Prolonged priming is actually counterproductive. And this scenario is scary because this is only talking about PGCs, but the paradigm might be different for other lineages. What about HSCs, blood stem cells? Over 40 years of research, in mouse stem cells even, nobody can make authentic HSCs to transplant them, which is very, very surprising. Therefore, our group has been really focusing on understanding these questions, but us as a field, we were stuck because we can convert naive and prime stem cells all day long quite easily, but I don't have a continuation and have a readout for this assay. If you think of this as an enzymatic reaction, I would like to have in a dish, A becomes B becomes C, and they perturb this reaction, but we don't have that. So what our dream was to really start with stem cells and have them make something like an embryo, which we now call a synthetic embryo model, in the dish. Now, this is quite ambitious because if you want to do it with stem cells, first you want to do this with natural embryos. It's not that we can take a natural embryo for post-implantation and grow it outside the uterus. So therefore, our goal was first, let's try to do this with natural mouse embryos, and if we're successful, maybe try to ask what happens to stem cells in these conditions. But I want to talk about a different spin of looking at this differentiation. Differentiation, as I proved, is, of course, there's a lot of success, but mostly a lot of difficulties. Typically, we direct differentiation scientists. If we want to make a cell type, we dictate which cytokine, which extracellular matrix, which conditions are provided at each time point. Now, you might argue, well, Jacob, your iPS cells and ES cells are not good enough, and that's why you don't make bone marrow cells. But if we take these iPS cells and ES cells, inject them into a mouse embryo, put them inside the uterus, they will make excellent bone marrow. So the problem is not the cells. The problem is us, the scientists. We're not able to mimic the sequence and the environment that exists in the context of an embryo in the context of an in utero. Therefore, this is basically a very good platform for differentiation that we would like also to establish. So we took a multi-step approach. Is first to try, let's make conditions to grow mouse embryos, natural mouse embryos. We take them out of the uterus and grow them for prolonged periods of time, and then use these conditions to take mouse stem cells and see how they would behave in these conditions. And of course, we want to move them to human stem cells and make human embryo model. And this is the path we took. And of course, the starting and the critical problem that we have with this research is really what's called the uterine barrier, which is in mammals, we can grow embryos until the blastocyst, and that's why IVF exists, but all these post-implantation stages, which are the most exciting ones, because as soon as the embryo enters the uterus, you will have gastrulation, which is the budding of the organs, and then organogenesis happens very quickly. And this happens inside the uterus in mammals, non-transparent, we cannot control it. Of course, if you talk about humans, organs are made already by day 40, but we cannot get human embryo samples. Women usually don't know they're pregnant, and even if you get samples here and samples there for making atlases, which is important. But if we want to study developmental biology, or if we want to use it as a differentiation, we need millions of samples. Therefore, we started thinking again about this ex utero embryogenesis. And therefore, the idea was, let's try to think of incubators, specific incubators, and try to slowly define conditions to push embryogenesis beyond day 4.5. And the idea is to have an incubator, a controller box, a gas mixing, and figure out the parameters. And of course, engineering. We ended up with a first prototype device, which really allows very careful control of where the embryos grow, as I'll show you, and the different conditions that are needed. And I want to say this is one example. We've improved this much than more, but started showing feasibility of this. So we've learned that you need to control different oxygen at different time point. Atmospheric pressures is very important, high glucose content. We have to develop the media, this yellow media in which these embryos are growing. And they like to grow in glass bottles. They don't adhere, because if they adhere, they get distorted quite easily. And a lot of details. And this is what it looks like. We've developed how to set up the system. These are the bottles. You have the media. How do we, what we call feed the embryos is basically we move them from the bottle every day, and they have new nutrition, and they get nutrition by diffusion, and they can grow and so forth. This is all now published, and I want to say available for academia, for research. And what I show you, if we start, for example, from day 7.5, and we use this protocol, it's very efficiently over 85%. You can start from a ball of stem cells that has no shape. Within 1 day, you get the yolk sac, heart beating, embryo turning. You have blood here for the heartbeat. This is the blood of the embryo. It has a placenta and a yolk sac, but it's not connected to the mother. And we can reach day 11 in development, ex utero. We can now reach day 13 in development, I want to say, which is really the embryogenesis, equivalent almost to day 80 in human embryos in terms of stage. And what also this shows you that what we can do as a research tool, we can start seeing embryogenesis really at high definition. We can look at specific organs here. You're seeing gastrulation. You see the embryo growing. This moving thing, this is going to be eventually the spine and the brain developing. So you can really see developmental dynamics at unprecedented resolution. So this was exciting and really showed us that the uterus is overall not necessarily needed to define the shape of the embryo, but it's a very good metabolic support. But the embryogenesis is basically intrinsically encoded within the embryonic material. Therefore, the next question was, can we make now embryo models from stem cells? Or in other words, now that we have a system, a condition that support natural embryogenesis, let's try to put stem cells and see how they would behave. And there, when we started approaching this, we learned something new when working with human pluripotent cells. Human pluripotent cells were thought that are not totipotent, that they cannot give rise to placenta and yolk sac. But when we work with humans, we saw that actually they can easily, much easier than in mouse even, give rise to trophoblast and primitive endoderm. And this is not really true. They are totipotent. They can do it. And therefore, what we thought is to do is really to start everything from naive stem cells, whether ES or iPS cells. Again, thinking in the humans, we don't want to use placental-derived materials, donated cells. We want everything coming from the initial source or the donor patient. And we want to take these naive cells, we want to split them into 3 populations. One we will nudge to become trophoblast, the placenta. One we will nudge to become the yolk sac. After 24 hours, put everything together, and then mount them on the same device, the same conditions we use for natural embryos. And really this worked in about 1% of the conditions. Now we have much higher efficiencies. Hopefully we'll report it soon. We start with 25 cells. The first 3 days you will think it's just a clump of cells growing. But if you look at day 4 of the protocol and you compare to a natural mouse embryo, you start realizing that this is an organized structure. You have the trophoblast on one pole, the embryo on the other pole, and as with every day you start realizing you have the structure, you have the primitive streak, you have the extraembryonic ectoderm. A day later, the amniotic cavity, the exocellular fold. Day 7, boom, yolk sac with an embryo-like entity, and that we can reach day 8.5 in development. And 8.5, just to show you here, is now you're looking at the same device that I showed you before when it's working, except that you're not looking at natural embryos, you're looking at thousands of synthetic embryo models that we can make each batch from their growing in these conditions. And you can see here, just show you, this is the brain development, there's the heartbeat, there's the valves, there's the umbilical cord that is growing, and we have characterized this very, very deeply, really to show you that this is compared to the natural embryo. You could see that you can have the structures, the heart, the brain, the neural tube, and I want to emphasize, this is self-organization. We do not give morphogens. This is all secreted by the stem cells themselves. So basically, this is a really self-organizing entity. And of course, we then moved to do this in human stem cells, and the challenge there, first, we were lucky because we've been working with human naive pluripotent cells, and thus we already developed them because these are the ones needed to give rise to all the tissues surrounding the embryos. And just to show you actually what we were trying to get, until the blastocyst mouse and human are nearly identical, but afterwards, huge difference in development. So now when implantation occurs, you can see now the blue part, the rooftop is the amnion, the blue and the yellow where they meet, this is the bilaminar disc, it doesn't exist in the mouse, and the yolk sac goes down, and it really, the embryo stays flat. And if you look at it from the top, you see it has a disc shape, okay? And then on day 14, you start to have primitive streak in the posterior part going, and that's how gastrulation really begins. And then gastrulation ends at day 21. So as you can see, this is different from the mouse. And I want to show you a different example, really very different from the mouse. And now this typical YouTube movie, you see this darkening, and then suddenly this orange extraembryonic mesoderm tissue appears, which is critical. Now, why is this black darkening? Because we do not know where these cells come from. So we cannot animate them. And it just shows you in 2024 how much we don't know about non-mouse development. So we try to mimic these stages, and I will not get into the details that are now published, but with similar optimizations, we start with naive cells. We can nudge them to become trophoblast, hypoblast, and really we developed an 8-day protocol that you get about structures that resemble day 14 human embryos only from stem cells. If we stain them, this is what I showed you in the video, you can see the blue here. This is the amnion. This is the bilaminar disc with the yolk sac. And this is all surrounded by the trophoblast. This is the chorionic cavity. And again, an embryo model, it's about structure. We have organization of the structure, not only the cell types. And we use light field microscopy really to look at, you visualize these embryo models. Again, the trophoblast on the outside. And soon you're going to see, just as I showed you, see here the amnion rooftop, the epiblast, the hypoblast, the yolk sac coming together. This is segmentation of an actual stage staining. This is not animation, by the way. So just to show you a couple of examples, what's nice about these embryo models is that once it happens, they are self-propelling, what we call developmental dynamism. They start having structures that didn't exist before. So if you look at the yolk sac, and we compare to an atlas of an embryo, we don't use natural embryos in this study, by the way, we just compare with atlases, you could see how actually the yolk sac, the epithelium here, the visceral one is columnar, and this one is more flattened. So really you get fine details of embryogenesis, again, appearing in this process when things happen right. And this is just one of the examples I showed you. In another example, if we look at structures, this is called the stalk. Again, doesn't exist in mouse. It holds the embryo. And the posterior part, you get brachyury, which is the beginning of the gastrulation. And here, this is the AVE, which is the other pole of the embryo. And again, you have anterior-posterior polarity. Last example, structure that doesn't exist also in mouse is these lacunas. So the trophoblast start having these empty windows that get filled with blood naturally. You can see we are peeking into them. So this is a lacuna. So again, just shows you the complexity that starts emerging from these cell types that's happening in these embryo models. So if we look at the summary of the details, we've learned about that naive pluripotencies are important. They're de facto totipotent. There's a lot of basic molecular biology questions that are important here. But we're also learning that working with natural mouse embryo and devising these conditions can teach us and help us how to grow stem cell-derived embryo models. And of course, we can make complete embryo models in mouse and humans and reach different developmental stages. And even in mouse, we can pass through gastrulation. And this is very exciting for us. And I want to emphasize why this is related, and this is something we do in a company I co-founded. So the embryo models has many, many promises, whether you're talking about understanding embryogenesis, whether you're talking about teratogens for embryo development. But this is actually a differentiation protocol because we're piggybacking on the ability of the cells to differentiate. And really, when we look at the quality of the cells that are emerging, are 99.9 similar to what you get from an embryo. And again, I think it's actually not surprising because there's a reaction, nucleation of embryogenesis that yields this, and things get secreted at the right time, at the right magnitude, which is what the embryo material happens. And therefore, there is an advantage you ask in this vision when it gets to the fields of aging or transplantation. Is it justified and is it reasonable in some cases where you can think of a patient, let's say, who needs a bone marrow transplant or a woman who needs her own eggs for whatever reason, that you can take with the adult consent to take stem cells, make naive stem cells, grow the embryo model for 30 days in case of the liver HSCs or 60 days to make an early gonad, and then take these cells and transplant them back to the patients. And these are cell types we haven't been able to make through conventional differentiation. So these are just two examples of many things that we do, and we try to look at and develop in our work to reach that with human cells. With that, I want to summarize and thank the different lab members that work on this and our collaborators, the years, and of course the conflicts of interest I've mentioned to my talks. Happy to take questions. Thank you.
Thank you. Very cool. Very cool. We've got time for 2 questions. So over there.
Thanks for that.
An amazing achievement.
So I was wondering, have you seen any evidence of the rejuvenation event that happens early on in embryogenesis around gastrulation?
Maybe the other one.
Yeah, I can repeat the question. I didn't hear. So I was asked whether we see.
There's something more.
Okay, so for the first one, do we see any signs of rejuvenations during what we see?
Yes.
So the answer is yes, because first of all, it is known also when you make iPS cells, whether you make it from an 80-year-old or 1-year-old, the product is actually embryonic, meaning even if telomere length, it gets elongated back to ground zero, which is happening there. So if you really see a lot of the markers, if we look at the bone marrow or the early progenitors at day, for example, 8.5, you can see that they're basically markers that are very similar to an 8.5 embryo, even if the iPS cell was made from a much older mouse, if we're talking about it.
Okay, so what about epigenetic clones? Because as you know...
Please stand up. Sorry.
Yeah.
So as you know, the Glanville lab found that at embryonic day 6.5, 7.5, the methylation age goes down.
Yes.
And then it goes up again.
Yes.
So if you saw that in your model, that's an interesting way to find the fact.
Exactly.
For that.
So one thing that we actually, we will not be able to do it without reversing the aging class, because I didn't get into it. One feature of naive pluripotency is erasure of DNA methylation. So it's not only about the telomere, the DNA methylation gets reset. Therefore, yes, the clock is reversed by fact because all DNA methylation, and this is what happens in the inner cell mass. And that's why we also put a lot of emphasis on doing things by going first through the naive pluripotency, because then you reset everything. Also, you reset developmental potential, and you reset ability to make trophoblast and yolk sac. You get all of this in one package, and then you do it. I think that really the more convincing answer that happens that I think once we reach day 11.5 and about 13.5, in the human day 28, because then the tissues are more mature, so we can really see more stronger phenotypes. But I want to say it's inevitable. You cannot do it without reversing the aging clock, because that's what happens with naive pluripotency. It's by definition a hypomethylated state.
Fantastic. Thank you very much. It was very impressive.